Thanks for waiting a little extra with the AV issue. So we're delighted today to have Dr. Christopher Walsh from Boston Children's Hospital for Somatic Mutations and Brain Disease 101. So this is part of the HBI 101 series. So Chris is chief of the Division of Genetics and Genomics. He's the Bullard Professor of Pediatrics and Neurology here at HMS, an investigator at the Howard Hughes Medical Institute, director and co-PI of the Allen Discovery Center for Human Brain Evolution, amongst many other wonderful titles. He has a PhD in neurobiology and an MD from the University of Chicago. And then for his internship, his residency, and chief residency, he came to the Boston area. He did postdoctoral research with Connie Sepko here at HMS. His research is very broad-ranging, but he's a world leader in studying the development and the function of the human cerebral cortex, and is famous for analysis of a variety of different genetic conditions that affect the developing brain, such as intellectual disability, epilepsy, autism spectrum disorders, and is expanding that work into the field of neurodegenerative disease work, aging, very broad, exciting work. And today's event is co-hosted with the Tan Yang Center for Autism Research here at HMS, run by Mike Greenberg. His lab is part of that. So if you're interested in learning more about Tan Yang Center, Jen Gibson will be here later today. And yeah, he has won many, many awards, but in the interest of time, I won't go through them all. Just in terms of housekeeping, you're signed up for the lunch, and if you have a confirmed spot, if you can come to the front at the end, and we'll walk over to Goldenson 229. Thank you so much, Chris.
Thanks, Parazad. Thanks, everybody, for coming, and thanks for being here online, those of you who are online. We'll be talking about somatic mosaicism in the human brain, and I didn't exactly know how the 101 lectures differ from the usual seminars, because it's a lot of the same audience. I'll try to give a little bit more background for those of you who might have less background in genetics. I'll be a little bit more sort of conceptual, but there will, and everything I'm going to talk about is either published or available in form on bioRxiv. So in some places I'll take the liberty of asserting things without showing a lot of data, because assuming it's sort of a more general audience. So I'll be talking about developmental, clonal somatic mutations that occur during the development. How do they form? Tell you how they represent a developmental map of the human brain. I'll talk a little bit about survival of the fittest occurring among neurons during development. And I'll give you a few pointers that I believe they're a major, largely undiscovered source of risk for diverse brain diseases. And I'll illustrate that with a few examples. And then I'll talk about age-related somatic mutations that occur after birth, after neurons, for example, are already mitotic. These can be clonal, they're in dividing cells like glial cells, or private, they're neurons. I'll show you how they measure age, cell type, gene expression, and cell exposures, and a little bit about how they change disease. So what are somatic mutations? Somatic mutations are mutations of the body or the soma. They are distinct from inherited variants because they're in some cells and not all cells. And the clonal somatic ones I'll be talking about arise during development in any one of the random dividing cells of the embryo, and they can form patches, or maybe they can be intermingled. They can occur early and be in a lot of cells, or they can occur later and be in a small patch, or as I'll tell you, they can occur after a cell stops dividing, and we now have the technology to determine that, in fact, even post-mitotic neurons have private somatic mutations all in the cell. So historically, this concept of somatic mutations was described in cancer. This is the classic, this is the sort of classic figure from Vogelstein that shows that these colon cells, first they acquire an APC mutation, and that clone grows a little faster, and then they get KRAS or BRAF mutation. That clone grows even faster, and then like P53 gets mutated, and the genome gets all sorts of damage, and it becomes unstable, and then you gradually progress to a tumor. So this progression of somatic mutations making cells progressively more malignant. But now we have the technology to study single cells. And we see that the picture is much more broad. So let me tell you about what types of somatic mutations I'll be talking about. I'll be talking about single nucleotide variants. So these are substitutions of one base, in this case a T, to an A. And I'll be talking about small insertions and deletions. This can be a deletion of 2 or 3 base pairs, or an insertion of 2 or 3 or 4 or 5 base pairs. These are generally small scale insertions and deletions. I'll refer to them as indels for short. And these collectively are referred to as point mutations because they involve just one or a small number of base pairs. And then there are the larger scale changes, chromosome aberrations, where you can get a gain or loss of an entire chromosome, or you can get large duplications and deletions. Topologically, these are the same as indels, but they're just really big. Instead of being just a couple of base pairs, they might be megabase size. They might delete or duplicate dozens of genes. And they arise by a different mechanism than the small indels, actually. And so these I'll be referring to broadly as copy number variants because they change the number of copies of one or more genes. Instead of the normal 2, you might have 3, you might have 4, or you might have only 1 if you have deletion. So these diverse types of mutation have diverse causes as well. The single nucleotide variants generally arise by some sort of chemical damage to the bases. And depending on what the cause of the damage is, you might have a substitution of a T for a G or an A to a C or whatever, and that depends on the chemistry of the damage and how it is misrepaired by the repair system. Indels, on the other hand, generally reflect a breakage of the sugar backbone of DNA, either a double-strand break or a single-strand break gets misrepaired so that instead of filling in the gap, you end up actually creating a gap on the other side. So you have a stable loss of a couple of base pairs. And then finally, the large scale changes, the copy number variants, these typically occur in divided cells only. And they represent some sort of miscopying of a large stretch or missegregation of chromosomes. So generally, these will occur in the cell cycle. And by the way, feel free to interrupt me with questions anytime you want. So like I said, our understanding of somatic mosaicism has really been revolutionized by the ability to study the genome of cells one cell at a time. And in our lab, that technology was developed and applied to neurons by 2 graduate students, Gilad Agroni and Shuyu Kai. They originally wanted to look at line elements or retrotransposons. And so they sorted single neurons using new end staining and fax machine, put 1 nucleus in each well of a microtiter plate, and then amplified them using a highly processive polymerase. And there are various technologies now where you can do this. This was just the original way. And then you sequence the genome of this cell as though it was the genome from your blood and compare it to the genome of that cell or the genome of that cell. And it turns out we went into this assuming they were going to be mostly identical, but it turns out everyone is completely distinct. And after that, we started looking at copy number variation, and then finally we looked at single nucleotide variation and these small indels. So this shows what happens for single nucleotide variants. Black represents neurons, red represents oligodendrocytes, and these are in most cases from the same donors. And so this is a series of postmortem brains from donors who died either right after birth or around age 20 or in middle age or as old as 100. And from each brain, then we took, in this case, 3 neurons from the dorsolateral prefrontal cortex and amplified them and sequenced them. And what you see in the case of neurons is an incredibly linear increase in single nucleotide variants with age. It's incredibly linear. It's very precise. It's exactly 17 or 18 single nucleotide variants per year, 17 or 18 in the first year of life, 17 or 18 in the 100th year of life. It's not exponential. It doesn't increase with age. It measures time. And exactly how the genome does that, how it creates 1 mutation every 2 or 3 weeks is a mystery. Oligodendrocytes, and the neurons, of course, are born with somewhere between 50 to 300 clonal somatic mutations that occurred in the progenitor cells of the brain. And so that's why the y-intercept is a couple of hundred, and then it goes up from there. Oligodendrocytes actually accumulate mutations faster. You expect them to, because after they divide at least a little bit, or at least are derived from OPCs that divide. But the surprising thing is that neurons actually accumulate mutations as fast as they are because they do not divide. And in fact, when you compare neurons to other cell types, they're kind of in the middle of the road. This shows this slope of that line for a bunch of different cell types. And in fact, the slowest one is sperm, and they divide. And the fastest one is cardiomyocytes, and they don't divide. So it turns out how fast you accumulate mutations really doesn't have to do with cell division. Neurons are kind of in the middle of the road. They're right at almost exactly the same rate as hematopoietic stem cells, which obviously have a very important role in cell division. And so exactly why it is that it doesn't matter whether you're dividing or not in terms of accumulating double-stranded single nucleotide variants with age is not completely clear, but it is a kind of again, surprising thing we're all still trying to figure out why do all cells accumulate mutations, and in such a linear way. All of these cells accumulate these mutations linear with age. Now, getting back to this cancer analogy, what happens here is that the cells then accumulate mutations. In the case of the colon, every cell accumulates about 30 mutations per year. But then, in fact, most of those mutations are functionally silent and have no impact at all on the function of the cell. They're deep in an intron somewhere or nowhere near a gene. But in the case of this clone, it happened to hit the APC gene, and that's why that clone starts to take over. It has a survival or a proliferative advantage. And then along comes a KRAS mutation and has a larger proliferative and survival advantage. And so again, that's how we thought of it as cancer. But in fact, if you study any epithelium in the body, it actually looks something like this, where in fact, there's a bazillion different clones that have various driver mutations in various cancer genes, and they're actually all competing in the case of this epithelium. So some clones are big because the mutation might be particularly forceful, or that mutation might have occurred relatively early in development, or both. And then other clones are small because maybe the mutation just occurred a couple of cell divisions ago. And so then these age-related mutations can be either neutral, as they are in most cases, and or they can, as I say, if they happen to hit the right spot in a driver gene, they can push the cell to become a dominant clone and to take over, and in some cases go on to cancer. But it turns out then that cancer, so there are studies that have shown in the epithelium of the breast in postmenopausal women, every single cell has a cancer driver mutation. Fortunately, most of them are not going to ever form a tumor, but the only cells that seem to survive are those that have cancer driver mutations, because the cancer driver mutation, they don't only stimulate proliferation, they also tend to immortalize cells and prevent them from the normal occurrence of normal programmed cell death. So again, the brain might be an unlikely place to be so obsessed about somatic mosaicism, and because of the fact that the neurons are all postmitotic. Just to review, in the case of the neocortex, the neurons are formed before birth, the earliest ones being formed a couple of, about 10 weeks after conception. And then the layer 2 neurons at the top of the cortex, they formed about 20 weeks or so post-conception, and they're formed in a sequence. So the deep layer neurons are formed first, and then progressively more superficial layers are actually formed at later gestational ages. But in the case of the neocortex, this is all done well before birth. Of course, we know there are parts of the brain like the hippocampus or the olfactory bulb that continue to form neurons after birth, but I won't be really talking about that today. Well, so we can actually see that development in any human postmortem brain that you care to study. And this was work done by two former postdocs, Mike Lodato and Molly Woodworth, who adapted that single neuron whole genome approach and then started looking at single nucleotide variants. And a more recent paper was published by August Wong, and then even a more recent paper was just in Cell Reports last fall from Sonia Kim and Vinay Viswanathan from Peter Park's lab. And so they analyzed, in this case, here's about 136 frontal lobe single neurons, and about half of them could be organized into lineages by the existence of, so every radius represents a single neuron. And then these ones all have this one particular variant that I'm just calling A1, these guys all share B1, these guys all share C1, and so on. And then within these broad branches, so what you see then is a patch of the cortex is a mixture of cells that diverge from the very first stages of development. And in fact, the pink cells in that patch of the cortex have more to do from a lineage basis to pink cells in the liver or in the heart than they do to neighboring blue cells, because they were part of different clades that diverged probably from the 8-cell stage. And then within the branches, you can see that there are, in some cases, additional single nucleotide variants that occur progressively later. So in this case, C2 is in some but not all of these cells, and 3 is in a smaller fraction, and so on. And then you can map those variants across the brain. So you see that the variants occurring early are all over the place. They're all over the whole body. They're in all of the tissues. And then the later ones are just in a little patch of the cortex right around where the cells were originally isolated from, because it's a much smaller clone. And then also, if you look at the cell types that contain them, and this is the work that August did, you can see that in fact the early mutations are in all of the layers, but then the last formed mutations are restricted to the middle and the upper layers, because these are mutations that occurred after the lower layers were already postmitotic, and so they're only found in the last born neurons, which occupy the most superficial parts of the brain. And again, the numbers of these mutations are quite large just from the single nucleotide variants alone. So this lineage map can be resolved to any level of detail that you want to, provided that you have unlimited funding to do that. So that's a little bit about single nucleotide variants. Now I'm going to talk about a different kind of variation, the copy number variants, and tell you about how they, what happens to them over the course of development. And this is work which is available on bioRxiv, but which is still in revision for publication. And so it arises out of work that was done in other labs. And this is actually work that's been contributed to by a lot of in vitro fertilization labs, suggesting that the blastocysts, the early cells of the embryo, are very error prone. There are a lot of errors of chromosome replication. So that in this case, if you take a blastocyst, you separate it into single cells and you do that whole genome sequencing like I just told you we did in neurons, you can see this is a normal one. And so this is the copy number on the y-axis, and then the different chromosomes are laid out on the x-axis. So this would be chromosome 1, chromosome 2, chromosome 3, and the X and Y chromosomes down here. So this one has got an extra copy of whatever that is, chromosome 18, let's say. And then this one is missing a copy of chromosome 21. And so these mosaic chromosome anomalies turn out to be very scarily common. And the IVF people, they will like genotype a couple of cells before implanting it, and they would frequently see vast, large-scale chromosome anomalies, and they didn't know if they should implant that or not. But it turns out that they are typically mosaic. And so this shows basically a couple, like 24 blastocysts, and every single one of them is mosaic for a chromosome anomaly. In some cases, most of the cells are abnormal, shown here in red. Typically, about a quarter of the cells have these large-scale gains or losses of entire chromosomes. Well, what happens to them during development? It seems like they would not be good for you in the brain, because gains or losses of entire chromosomes we know is very bad for neurons. And so Diane Shao, a pediatric neurologist, worked with Yifan Zhao and Peter Park's lab to sequence a whole lot of neurons from the fetal brain. And you can isolate fetal neurons because they collect in the cortical plate, which is almost 100% neurons. And then they sequence them using a high throughput sequencing technology that I won't describe in the interests of time. And so this shows now the chromosomes are arranged around a clock, and each circumference is the genome one single cell. And these are what the prenatal ones look like. That's what the postnatal ones look like. And then red is gains of chromosomes or segments of chromosomes, and green is losses of chromosomes or segments of chromosomes. What you can see maybe is that the prenatal ones, there's a lot of stuff going on. There's a lot of green and red, whereas the postnatal ones look much more normal. And to quantitate that, here is it. Diane actually, at the demand of the referees, did this whole experiment 2 whole times over using 2 completely different methods, one based on whole genome sequencing using polymerases, another using the TN5 TN5 method, and you see essentially the exact same thing. The prenatal neurons, about 25 or 30% of them have gross, large-scale gains or losses of almost a chromosome. And then postnatally, it's about 5%. So there's a huge loss. And this is what the individual cases look like. The prenatal ones are all over the place. There's a lot of inter-individual variation, and that's probably because the creation of these mosaic things is very random. But then it's very consistent after birth, with about 5% of the neurons having it. So what happens then? Well, as far as we know, there's no mechanism to correct a CNV or replace a chromosome. And so the plausible mechanism for this disappearance of these grossly abnormal neurons with grossly abnormal genomes is that they die. And in fact, we know that there is death of 20 or 30% of the neurons in the cerebral cortex. That's been known for decades. And this is an example of it in mice. Here is E17 right around the end. Shows the hippocampus. And so this is around the end of formation of the pyramidal neurons of the hippocampus. And then this is staining for activated caspase-3 to highlight dying cells. And you see that there are few with E17, and then there are more at E18, and then there's a ton of them at E19 and right around P1, and then they're all gone. By a couple of days postnatal, that cell death that wave of cell death has passed. And it's always been a mystery. What's going on with this cell death? Because they're not limited to a layer. They don't like relate to the mapping of the hippocampus or the structure of the hippocampus. It's just like scattered cells all over the place. What's the logic of that cell death? And this similar process seems to happen in the human neocortex at the end of gestation, basically the second half of gestation. Well, there's been a lot of study of programmed cell death of neurons, and the history of this goes back to Rita Levi-Montalcini, the discovery of nerve growth factor, and the fact that regulates the survival and the death of neurons. And then Barbara Finley in the 70s applied this to the cerebral cortex, and she described this, again, random loss of neurons all over the place, and she sought for some sort of logic to why some of the cells die. And there are more recent papers. This is the lovely paper from Oscar Marin's lab showing this 20 or 30% loss of both the excitatory neurons and the inhibitory neurons, and showing that it depends on the formation of synapses. So what was clear, even back going into the 70s, is that neurons have to make synapses in order to survive. And that if synaptic formation is blocked, this cell death increases. And if you have overactive synapses, you can actually rescue some neurons that would ordinarily die. In fact, this goes back even further. Almost 150 years ago, Johann Bernard von Guten, a psychiatrist, first discovered that neurons have to make synapses in order to survive. Now, von Guten is best known because he was the personal psychiatrist of King Ludwig II of Bavaria, who in turn is best known because he built the crazy Neuschwanstein Castle, upon which the Disneyland Castle is based. King Ludwig spent so much money building Neuschwanstein and other castles, and he had a lot of other very eccentric practices, that the government of Bavaria came to von Guten and said, we need you to declare him insane. And so he did do that, and he was forced to abdicate. And the two of them went for an evening walk 2 or 3 days after his abdication. And they disappeared, and their bodies were discovered the next morning floating in 3 feet of water. And exactly how they died is a complete mystery. Whether it was an assassination attempt, whether Ludwig tried to kill von Guten, who knows? But before his untimely demise, von Guten was actually a noted neurobiologist. He studied people who had lost one eye and then survived for years after that. Here's one of his pictures published posthumously by his son. And it shows this missing eye, and this is the lateral geniculate nucleus just on the surface of the brain, and this is the first relay of the eye. And the larger projection goes contralaterally, and perhaps you can appreciate that the contralateral and that lateral nucleus is shrunken relative to the one on the same side. And it's clearer if you section it, because the lateral geniculate nucleus has these layers, and it's specifically the layers of the lateral geniculate nucleus that ordinarily receive input from the eye that's gone that you can see they've died. And in fact, for decades, this was how the anatomists traced connections in the brain. They would damage one step, and they would look at and they would say what cells are dying across a synapse. And so this is death across a synapse. These lateral geniculate neurons have not been damaged. They have just lost their synaptic connections. They have to maintain their synapses, otherwise they die. And so this is a fundamental property of at least most of the neurons in the brain. In fact, if you take out the eye prenatally, some of the visual cortex neurons will even die. So we wanted to know whether this loss of neurons or this loss of copy number variants was actually coming about because of this process of programmed cell death and whether it related to formation of synapses. And so Diane first asked whether you could find the copy number variant containing neurons in cells where you also could analyze their RNA gene expression pattern. And so by using single cell ATAC-seq and RNA-seq, in fact, you can call copy number variants, large ones at least, and it's not as sensitive as the sequencing methods. And so the rate of it prenatally is lower. Again, you see it's highly variable, but there is again more prenatally, and then again, about 5% of the neurons that have these large scale copy number variants postnatally. And so now this is great because it allows us to ask what's going on with the transcriptome of these neurons that carry large copy number variants and that we know are fated to die. Well, it turns out they fall into two categories. So that if you look at the transcriptome of copy number variant containing neurons, there are those that express low levels of synaptic genes and high levels of cell death genes. And then there are others that express low levels of high levels of synapse genes and low levels of cell death genes. And so in fact, some neurons look like they're forming synapses and they have turned down their cell death pathways, and other ones look like, in fact, they might be fated to die because they have high expression of cell death genes already. And this shows this separation for the excitatory neurons. You can see the sample size is smaller, but you can see a trend among the inhibitory neurons also that they're falling into these two categories. And here is those two categories again writ large. You can look at the pathways that are upregulated. These are the copy number variant containing neurons that have low synaptic gene expression and high cell death gene expression. And they form a category. And you can see that this yellow category where there's copy number variants and they express cell death genes, but not high levels of synapse genes, they're very common prenatally, and then they become progressively less common after birth. So next we wanted to ask, what drives this loss of these damaged neurons? And we hypothesized that it was likely to be synaptogenesis. And so for that, Diane analyzed hotspots and cold spots. And so you can analogize that with this cartoon from World War II where they looked at fighter planes, where the bullet holes were in the fighter planes that successfully returned home. And these places where there are lots of bullet holes are the places where a bullet hole is consistent with still being able to fly the plane home, whereas they never found bullet holes in these cold spots because a bullet hole in the engine or in the pilot in the cockpit was not consistent with being able to successfully navigate home. And so we asked, what are the cold spots in the genome? Namely, those parts of the genome that are never deleted in neurons that are alive, because those are the parts of the genome that are essential for neurons to survive this process. And so cold spots tend to have a lot of genes in them, as opposed to parts of the genome that are gene poor, where it might be expendable. But also not just any kinds of genes. They are enriched in genes that are associated with developmental disorders like autism. They're enriched in genes that are essential to put the synapses together. And so what we think is that these large-scale copy number variants occur during the process of neurogenesis into the dividing cells, because these copy number variants, as far as we know, only occur in dividing cells. We have more evidence that I'm not showing you. But then they're eliminated during the process of synaptogenesis. And this process of programmed cell death is particularly active during the second half of gestation. It probably continues onward, and we've got a lot more to say to that in the paper. It may be something which continues at a slower level actually throughout our life. Now, what would be fascinating to think about then, what happens if you have a condition like autism or intellectual disability where your synapses don't work for all of the cells? Does that disrupt this competitive process? And if it did disrupt that competitive process, your brain would be left with a lot of noise. It would be left with 20 or 30% of the neurons that don't have normal genomes, that are missing large segments of the genome. And so that's what Diane is looking at now to see, in fact, if this process relies on germline, on a normal germline genome as well. If there's an interaction then between germline mutations that actually might regulate this elimination process. Okay, well that's one story. As I told you before, you can have mutations that occur early, like this red one, and that are shared in the blood and the brain and all different tissues, but only a fraction of the cells. And then you can have mutations occurring later that are restricted to the brain, like this yellow mutation, because they occurred in the neural tissues after it's separated from the non-neural tissues. And then we can have mutations in little fractions of the brain. And so, again, most of these mutations are functionally neutral, but when they actually disrupt a key gene, they can cause a lot of different diseases. And I'll just mention a couple of them briefly. We see activated specific mutations in this mTOR pathway in focal pediatric epilepsy, mutations in the RAS-RAF-MAP kinase pathway in adult temporal lobe epilepsy. There's a contribution to autism by both mosaic copy number variants and single nucleotide variants I'll describe briefly. I'll talk a little bit about schizophrenia. So I'll just present a little picture of some of the diseases that can be caused by these. So our work on this really was started by Ann Pedori, who was a postdoc now 20 years ago and was interested in focal epilepsy. And one of the most amazing kinds of focal epilepsy is this condition called hemimegalencephaly, where half of the brain is grossly enlarged and is intensely epileptic. And this shows the MRI scan of this. And what happens is the surgeon will remove that entire hemisphere. So that here you see that the hemisphere is gone and it's just cerebrospinal fluid in its place. And this can be very effective to control the epilepsy. In the case of this patient, there were intractable epilepsy, 100 seizures a day starting at birth. The left side was weak because this right hemisphere was no functional good anyway. It was taken out and the patient was seizure-free for 6 years, learned to walk, developed fluent speech. He reads at grade level. And here he is. His name is Dante, and I use this picture in his name with his parents' permission. So she thought maybe these are like cancer mutations, but these aren't cancerous. These hemimegalencephaly doesn't continue to grow. It's a stable malformation, but maybe it's somehow related to a mutation that's just in that part of the brain and not the other part. And so she, in fact, showed one of the first mutations was an activating mutation in a gene called AKT3, which is part of this mTOR pathway, and work from other labs implicated genes all up and down this pathway as causing not only these large malformations, but also the much more common tiny malformations, which are called focal cortical dysplasias. And this is now canon in the epilepsy field, that these lesions represent clonal, somatic, activating mutations in the mTOR pathway. This is the most common cause of pediatric epilepsy that comes to surgery. We do it all the time over at Boston Children's Hospital. They're stable malformations, and they represent a developmental overgrowth, but then one which is stable. And if you use the most sensitive sequencing technologies, you'll find some sort of activating mTOR mutation in 80% of these cases. And this is work that was continued by Alyssa Tagamma. And it's amazing, you can get intractable epilepsy with fewer than 1% of the cells carrying the mutations. Typically, these lesions have the mutation in 1 or 2%. But it's also allowed the repurposing of mTOR inhibitors developed for cancer to now treat epilepsy. More recently, Sattar Kushku, an epilepsy fellow who now has his own lab at the Brigham, studied mesial temporal lobe epilepsy. This is the most common form of epilepsy in adults, and it's classically genetically complex. It has no germline causes as far as we know, and it's been known for 75 years. Nobody knew why, but if they knew that if you took out the temporal lobe, you could cure the epilepsy in 3/4 of well-selected patients. And in fact, when you deeply sequence them, they also have activating cancer mutations in that tiny fraction of cells, but almost never in the mTOR pathway. In this case, it's almost always in the RAS pathway, most commonly this gene called ATP11. And we think that these mutations arise during development, although in this case, the ongoing neurogenesis in the dentate gyrus of the hippocampus may actually modify these mutations throughout life. And that might be why the epilepsy doesn't occur as a newborn, but occurs typically 10 or 20 years later. So what about autism? In a study published now some time ago, Elaine Lim analyzed a whole exome sequence from blood. And so this would only identify the early occurring mutations, like that red one I showed you that would be in the blastocyst stage. So it would be in all of the tissues of the body in a fraction of those cells. And if you look at all of the de novo mutations that a person has, usually when they're heterozygous, they will have what we call a variant allele frequency of 50%, meaning they're in half of the DNA reads because they're heterozygous. There's a good copy and then there's a variant copy. But she found that, in fact, there's a minority of them that have this lower variant allele frequency, and these are the mosaic ones. And so she analyzed almost 6,000 families that were trios, where 2 parents and a child. And again, we have relatively low sensitivity to detect these because it's relatively low coverage sequence, but we have a large sample size. And we found that there was, and actually we did this simultaneously with work from 3 other labs, and we all found similar things. We all found an excess of these mosaic mutations in cases compared to controls. Various estimates suggest about 5% of kids with autism have a somatic variant that's likely to be contributory, although it's really too early to take this to clinical sequencing. Somewhat more recently, about 5 years ago, we published a study where we analyzed postmortem brain of autistic patients. We only, in this case, analyzed 59 cases and 15 controls. We did much deeper sequencing, 300x coverage, so we could detect a broader array of clones, and especially focusing on those in the brain. And we found a surprising increase in actually mutations that are in parts of the genome that represent highly conserved neural enhancers. And there's nominally more than a 10-fold excess in autism cases versus controls. We also found that the mosaic mutations in autism were more likely to be close to these non-coding regulatory mutations were more likely to be close to a brain-expressed gene, and some of them actually altered function. And we don't know why there's this, it increases these regulatory mutations, but this seems to be a consistent finding. In fact, a later study in schizophrenia, we increased the sensitivity a little bit by sorting the neurons and making DNA just from the neurons to focus on these early occurring developmental mutations. We found actually there's a non-significant overall increase in somatic mutations globally in schizophrenia compared to control, although we'll need a larger sample size to know whether this is a significant difference or not. But again, we find this enrichment now right at transcription factor binding sites, either in promoters or enhancers, in cases compared to control. So this is a disease-specific enrichment in regulatory mutations. And not just any regulatory sequences, specifically those regulatory sites that are active during the development of neurons. And furthermore, we could find there are 2 particular patterns of DNA damage. There are CPG, so a C to an A change or a C to a G change. These are called CPG transversions. And again, they're enriched right at within 100 base pairs of the transcription factor binding sites. And we think we know what these are caused by. During the early fetal development, there's a wave of demethylation of the genome and then remethylation. And the demethylated cytosines are very vulnerable to being chemically modified. And so we think that these mutations occur in some cells right in these first few cell divisions. And in fact, they're present in 15% of the cells or so, which is very consistent with them happening at the stage when there's only like 8 or 16 cells. So they're probably present also in all tissues. And then we found a second pattern, which are T to G mutations, also very highly enriched at transcription factor binding sites. And in fact, schizophrenia overall carries more T to G mutations than normal. We don't know what causes them. They are in a smaller fraction of cells. I mean, they probably happen later. And the 2 patterns of mutation are in 2 different cases. So some schizophrenia cases have the T to G, some have the CPG, and most of the schizophrenia cases have neither. We hypothesize, but we don't know yet, that these T to G mutations might be due to some sort of maternal exposure, perhaps some sort of maternal immune activation, or maybe a maternal infection, because these processes have been implicated in the causation of schizophrenia. And I'll just show one slide on ALS and FTD, where we think also these clonal somatic mutations have a role in disease risk, that we show that mosaic mutations overall are enriched both in ALS and in FTD in cases compared to controls. When you study postmortem brain, that suggests that they contribute to risk. And then when you look at where these mosaic mutations are, these are 3 different cases of all of ALS, and you see that the mutation is present basically only in the motor cortex, or in this case, motor cortex and parietal cortex. So these look like those little focal cortical dysplasias, where there's just a little patch of mutant cells. But in this disease, it's not a focal disease. It's not focal epilepsy. This is a disease that spreads through the whole brain. And pathologically, these brains look indistinguishable from the typical form of ALS. So this suggests that there must be some sort of pathological process that starts from a little cell in the brain and can spread through the spinal cord and to the other half of the brain. So in the last, I don't know how many more minutes I have, maybe 5, maybe a little bit more than that. Maybe not. I'll return to this age-related accumulation. I mentioned at the start that neurons accumulate SNVs at 17 or 18 per year, and oligodendrocytes accumulate SNVs faster, about 26 per year. But in fact, the neurons accumulate with these indels, these short insertions and deletions that I mentioned at the outset. Neurons accumulate them faster than oligos. And so every cell is different in the way it accumulates mutations and the patterns of damage and the patterns of nucleotide substitution. And neurons and oligos from the same exact spot in the brain have different patterns of mutation. Mutations in neurons are, since they're not dividing, it seems to be driven by transcription. Parts of the genome that are highly expressed are enriched for mutations, whereas parts of the genome that are not expressed are depleted for mutations. In oligos, in fact, the mutation seems to be driven by cell division. So it's part of the genome that replicate late are more vulnerable to these replication mutations and are more likely to be mutated in oligodendrocytes than the parts that replicate early. So I don't have time to mention all the people that have done this work since I'm running out of time. Well, I will mention this. So Zenon Joe Luquette from Peter Park's lab, Lan Dong from Alice Lee's lab, and Peter Park, and then also Chinsia Ma and Lan from my lab have collaborated with Mike Miller's lab and Ann McKee. We've studied a bunch of different degenerative diseases and asked, how does this pattern of accumulation of single nucleotide variants and indels, how does it change in degenerative diseases? And so this shows 4 different degenerative diseases: ALS, frontotemporal dementia, Alzheimer's, and chronic traumatic encephalopathy after playing American football. And in every one of these conditions, you see something fairly similar. The normal age-matched controls are these gray dots, and then ALS, FTD, they all show a little bit of an excess of single nucleotide variants, a modest excess, 100 or 200 extra single nucleotide variants compared to age-matched controls. It's a little bit, it's a little noisy. Some have more than that, and some look almost normal. And so in the diseased brains. We discovered this first when Mike was still in my lab or working with August Huang in Alzheimer's disease. So this shows the age-related, the increase in Alzheimer's compared to control. Again, it's about 200 mutations, equivalent to 10 years of aging at 17 SNVs and 2 or 3 indels per year. And that's just about how many years of life are taken away from someone who has Alzheimer's. We see that when you look at the pattern of nucleotide substitution, which suggests potential cause of these mutations, you see that in fact the one pattern which we call signature A is relatively unchanged, whereas a different pattern is rare in the normals but is highly enriched in the Alzheimer brains. And the signature A happens to be these red C to T changes and green T to C, whereas the signature C has a lot of these C to A changes. So it's a completely different pattern, as though a new mutational process is superimposed onto the normal aging. And we think that this has to do with reactive oxygen in the neurons that typically causes these C to A changes. Now, more recently, in work that was just published in a paper in Science last fall, in the case of CTE, and which is still online only in bioRxiv in the case of ALS and FTD, we've seen that in addition to the single nucleotide variants, there is this colossal change in these short indels. which are rare in the normal neurons, and they increase only 2 to 3 per year. But some of the neurons look completely normal, but some of the disease neurons have hundreds or thousands of extra indels, which is equivalent to hundreds of normal years of aging. And these are very damaging mutations. And so this looks like a more catastrophic process that affects some cells a lot and some cells relatively little. And they're not just any indels, they're specifically 2 base pair deletions, which this shows different kinds of indels depending on whether it's a 1 base pair deletion or a 2 base pair deletion or a 3 base pair deletion. And you see that ALS and Alzheimer both show this massive peak of specifically 2 base pair deletions. And that suggests a very specific cause. And we think we know what that cause is. There's topoisomerase 1, which is ubiquitous in neurons and regulates the folding of DNA, and which is essential for transcription as well as the removal of RNA that gets falsely incorporated into DNA, causes these 2 base pair deletions. That's well documented. And in fact, it creates a covalent complex with the DNA along with a 2 base pair deletion. They typically cause a single-stranded 2 base pair deletions that are normally repaired, but if left unrepaired, you're left with a 2 base pair gap on one strand relative to the other. And in fact, that's a bad thing to do. That fragments your genome. And in fact, Xenon has shown directly that in fact, the neurons, the brains of those individuals that show a lot of these 2 base pair indels, if you just run the DNA on a gel, you can see that it's fragmented. So the red dot indicates individuals that have a high rate of these 2 base pair indels. And if you just run it normally, you see a little bit of a smear.
I see.
You see a much better. And so what's happening in these diseases is that these indels are literally cutting the genome apart so that it becomes, so presumably makes it unable to transcribe properly. And so now we're thinking of neurodegeneration as a genomic disease where you have this oxidative damage that creates these SNVs, and that initiates a repair process. But in fact, this repair process can kind of run out of gas and engage this topoisomerase 1, which is an alternate repair pathway, but which creates these 2 base pair deletions. And that literally causes a lot of problems with transcription, but also literally causes the genome to fragment and to fall apart. So to conclude, the neuronal genome is a completely dynamic. It's accumulating SNVs and indels every year in a remarkably linear fashion. How does our genome record time is a complete mystery. They are enriched to transcribe parts of the genome. And all of the diseases we've studied so far show abnormalities in this pattern and this rate of accumulation that look like they're referable to specific processes. So like I said, the clonal SNVs are probably a major source of undiscovered risk for almost any disease for which we don't understand all of the risk. I used to think of them as like a little add-on to germline mutation. Now I think of them as actually the essence of disease, especially age-related disease. And it's in fact the germline genome that modifies what somatic mutations do. So the neuronal genome is constantly changing. And again, the genome it's a tablet. It records the life of a cell. Just as our genome records our evolutionary history, the genome of an individual cell records that cell's history. Every cell division that led to it is recorded by a clonal somatic mutation. But then what happens to that cell? How old that cell is, what sorts of environmental exposures it's had, what sorts of metabolic processes it have. It is a form of epigenetic memory because many of these mutations occur in transcribed parts of the genome. They're enriched there, and so they alter the transcriptional pattern of that neuron. And so they are a type of memory. So this gives me a chance to try to go back and capture the people that I forgot to acknowledge. The fetal CNV work I mentioned, Diane Shao and Yifan Zhao. The glial mutations were done by Javier Gantz and Sarah Bizotto, as well as Joe Luquette. CTE mutations, I think I captured everyone there. So I hope I've left a little bit of time for questions. Thanks very much.
It's really wonderful talk. It's likely the prominent age-related mutations accumulated, especially in oligodendrocyte, in glial lineage. There are astrocyte, microglia, so other glial cell types in the brain. But I don't know why it's more specific to the oligodendrocyte. Do they have some kind of protective mechanism? Astrocyte, microglia may have more protective mechanism to escape from removing those mutation-accumulated cells.
Well, we haven't studied the other cell types in the brain, like astrocytes and microglia, yet in the same level of detail, just for technical reasons, because we hadn't figured out how to purify them. But we do have preliminary data on those other cell types, and they look like they accumulate mutations. Again, every cell is different. We looked at inhibitory neurons, for example. They accumulate mutations maybe slightly faster than excitatory neurons. Astrocytes look like they might be a little faster than oligos. Oligodendrocyte precursor cells are faster than oligodendrocytes themselves. Microglia, I can't remember, but I think they're a little bit faster also than the oligodendrocytes. So every cell accumulates at least 15 or 20 SNVs per year, probably more.
So we have a question in the chat from Elizabeth. Does this research always, rely upon brain tissue samples? Can you see ways around brain tissue sampling, for instance, CSF samples or other sources of cellular material for diagnosis?
Well, so the dependence on brain sample, I showed stuff that depends on brain samples. There's lots of interesting things you could do that would not rely on brain samples, but it's, the questions that we wanted to ask with this technology required us to look at brain samples. We really wanted to look at the disease the way it is in somebody that died of that disease, rather than a model which may recapitulate it perfectly, but which might not. And so, then we feel like once we understand what mechanisms we want to further interrogate, then we want to take that into mice or into cell culture. I mean, iPS cells in culture, then, they will have their own somatic mutational history that will reflect the history of those iPS cells. They'll be accumulating a couple of SNVs every cell division. They also, depending on the tissue culture conditions, whether it's atmospheric oxygen or low oxygen, they'll be accumulating C to A changes, because cell culture creates C to A mutations when it's done under atmospheric oxygen. And so there's some things but there's really cool experiments that can be done in cells, in cell culture as well. And we're now also trying to look at other animals, compare this process. Do mice accumulate mutations at the same rate as humans, or do they die with the same number of mutations as humans because they accumulate them faster? And what about primates and so forth?
Do you find anything there, like in terms of looking at mice or primates or any other species that they accumulate mutations in a similar way?
Yeah, so that work is not on bioRxiv yet, but so far we looked at a couple of non-human primates, chimps, marmosets, macaque, and then also mice. And it looks like basically in all those species, they accumulate mutations at 15 or 20 SNVs per year. So mice accumulate them at the same rate as humans, but then they die with almost none. They only die with like 50, 80 SNVs.
So the rates are still similar, because what
This, by the way, is the opposite of what has recently been reported in the colon. So there was this beautiful study from Alex Kagan, who's now at the Sanger Center, where he looked at colon epithelial cells from 23 different species: whales, naked mole rats, mice, you name it, a whole zoo of animals. And in his paper, he suggested that the rate of accumulation might actually set lifespan, because mice, in fact, accumulate mutations in their colonic epithelium 30 times faster than humans. And so they die with the same number as humans. We both die with about 4,000 or 5,000 mutations per cell in our colonic epithelium, but mice get there much, much faster. And then whales accumulate them slower. Naked mole rats who live much longer than mice but are about the same size, they accumulate far slower than mice do. And so there's a fairly direct relationship between rate of accumulation in the colonic epithelium and lifespan. So we assumed that neurons would be similar, but it looks like, in fact, they're kind of the opposite, and that the rate seems to be constant across species, probably defined by the inherent metabolism of neurons, but then probably not so relevant for setting the lifespan.
Just to add to that, I mean, is there a way then to tie the causality? I mean, while we make these observations, science is always at the burden to show that you could reverse or stop, and it's causal.
Right. Well, yeah, it's hard to know. That's a very difficult argument to make because I guess because of the fact that, I mean, it's not my field, but also because people die of diverse causes. I mean, what is striking to me is if you look at age-related diseases, cancer is obviously driven by somatic mutation. Now, maybe, if you believe me, maybe neurodegeneration has a large role for somatic mutation as well. And we have other work that actually goes into that in more detail. And so if somatic mutation is ultimately the cause, if some somatic mutation-based condition is ultimately the cause of death, then it kind of makes sense that they would drive that somehow. But, exactly, it is controversial in the field just to define what is the hypothesis and how can we set up the experiment to test it.
Here's how like glial cells in the brain, if it's known, react to the neurons changes in single nucleotide variants or other genomic changes.
So yeah, we haven't looked at that because we've just been kind of trying to define the landscape first. But you can imagine that all kinds of things might happen. Some of these SNVs may create a neoantigen. It might make just that one neuron like now reactive to T cells or something like that. So let's see. I don't really think we have any information on exactly how this works. We think that we have a paper describing somatic mutations in microglial cells in the brain of Alzheimer's disease. And what we find is that some of the microglia accumulate cancer driver mutations, and those microglial clones tend to expand, and that process is accentuated in the Alzheimer brain. Those somatic mutations actually change the function of the microglia that carry them. They make those microglia reactive, logically reactive, and hyperactive, and also proliferation. And so they create a process in the microglia which is likely to be toxic to the neurons.
So I was really interested in your neurogenesis versus synaptogenesis and how neurons are getting pruned based on that distinction in their differentiation state. Do you have any hypothesis as to what's jump-starting the actual pruning of the neurons? Is it a population base, or when the synapses form, it triggers something inside the dysfunction neurons to go apoptotic?
No, that's a great question. So we've thought about that a little bit in our own experiments to see what pathways are activated at the transcriptional level in the CNV containing neurons that are also turning on their cell death pathways. There's also studies from animal models that have looked, this program cell death has been around for decades, and some people have tried to get at that. And I believe that I don't know a lot about this, but I believe that there is basically there's all of these calcium-mediated pathways that are downstream of NMDA receptors. And I think that there's a connection. If you have, I guess, insufficient stimulation of NMDA, you can have a calcium pathway that induces cell death. And so that's likely what is causing this, that there's a competition for synapses, and then those neurons that form insufficient synapses activate the cell death pathway. Might be directly connected to the synapses, but I'm not the expert.
So still 5% of neurons carrying the large CNV mutations survived postnatally.
Right.
And do they actually functionally incompetitive compared to normal neurons, for example, in terms of gene expression or in terms of synaptic transmission?
Well, so we don't know directly because we need to set up an animal model to do that. And again, I think in part of bioarchive, what we also find is that we also find neurons that contain smaller CNVs. And so those are not so much eliminated in that wave before birth or near birth, but in fact, smaller CNVs neurons containing smaller CNVs continue to be eliminated throughout life. And so this pruning process looks like there's a wave where the really sick neurons lose out right away. But in fact, it looks like this competition continues throughout our life. Like you guys, when you leave the room, you'll probably be short a couple of neurons that have small CNVs. And that might be an inherent part of the learning process. But also that shows that the neurons inherently have the brain has an inherent mechanism to eliminate faulty neurons. And that's really all you need for neurodegeneration to happen. If you keep hammering the genome and damaging it as you get older, then in the case of many neurons, the level of damage is likely to reach the point where this natural process of competition-based cell death may.
We have another question on Zoom. Jin asks, may I please ask if the neurodegenerative disease is related to genomic DNA damage repair problems? Will it be too late for the treatment by the time it is diagnosed, mostly for sporadic cases because the mutations have accumulated a lot? Have any proposal for therapy based on this genomic hypothesis?
Yeah, so we don't really know what the dynamics of these mutations are. We're capturing the, we're looking at neurons that were basically still alive when the person died. They still express NUN. The neurons that are already highly apoptotic don't express the antigens that we use to sort them. And so their genomes probably look way worse. We're looking at basically the neurons that we're showing you in disease are probably the best that's left, the least damaged ones. We don't know what the dynamics of the process is. Ordinarily, the process of repair is an ongoing thing. And so you would think that if you stop the damage, the neuron might have the capability to catch up. We suspect that having said that, we think that the repair processes might be deficient because of lower levels of expression of some of the repair enzymes. And so it might take longer to catch up in the disease state than normal. We like to think that there might be some therapeutic targets in there, but we're not sure. TOP1 itself, we're not sure whether you want more of it or less of it. And so we're not exactly sure what that means in terms of developing it there.
Thank you so much. I think, we can take one last question.
Yeah, or someone want to. Thank you for the nice talk, first of all. I have a question that is connected to another one, because I don't know his name, but it was saying you that maybe these neurons that survive and we can find in neurodevelopmental or neuropsychiatric condition are incompetitive. But to me, the hypothesis was kind of different, because is it possible that like the mutations that actually survive, let's say, of the neurons that then survive and do not die, renders them hyper-competitive? So they win initially the competition because, for example, they push forward synaptic formations. The neurons that don't survive underexpress the genes connected to synaptic formation, for example, or plasticity. But like the pathway that you were mentioning, like mTOR pathway, seem to be hyperactive.
Right.
So could it be that like these neurons win initially the competition in the first wave, then they form lots of synapses, and then afterwards it's starting a different kind of pruning that kind of removes this hyperformation of synapses, and that's why the symptoms then next start emerging. I don't know if it's possible. I think they found something similar even linked to Alzheimer's disease. So the question is that, could we define whether a SNV is going to survive or not, basing on the effect of this on synaptic formation and plasticity?
Yeah, that's a really fascinating issue and question you raised. I mean, I'm just very suspicious that these genetic causes of focal epilepsy that I mentioned, activation of mTOR and activation of the RAS pathway, not only do they make the neurons hyperexcitable, but they also immortalize them. So they would also exempt them from this competitive cell death process, because even if they weren't good at making synapses, or even if they made abnormal synapses, they have a tumor oncogene that's going to keep them alive. And so we might find that, in fact, this might be a recurring pattern in other mosaic diseases, that a lot of them actually are mutations that immortalize the cells. So it actually makes me wonder, it makes me look back at our mosaic studies of autism and schizophrenia, because most of those are, well, actually the autism at least, a lot of those are loss of function mutations. And so I would imagine that actually in the brain, a loss of function mutation in an autism gene, again, those neurons might just be lost. They might be lost in the competitive process. And so maybe we can detect the mutation in the blood, but it might not be so detectable in the brain. And so there's a whole dynamic of, it's not just creation of mutations, but this filtering process due to this kind of struggle for survival that we're just trying to get our heads
Thank you so much for the wonderful talk. So you've signed up for the lunch, if you can come here. And in case you get separated, we're gonna walk over, but in case you get separated, it's Goldenson 229.